Okay, here we go. So you guys, our first example of nitrogen fixation will be in cyanobacteria. Now, what's really important to know about nitrogen fixation is that very few organisms can perform it. Nitrogen fixation is taking molecular nitrogen and converting it into ammonia. Now, molecular nitrogen, it's two nitrogen atoms with a triple covalent bond. Very few organisms have the enzyme to break the triple covalent bond and reduce the molecular nitrogen to ammonia. To do that, you have to have an enzyme complex called nitrogenase. So again, very few microbes make nitrogenase, but we'll be looking at some of them. Now, one group of microbes that can make nitrogenase are the cyanobacteria, and the one that we've looked at already, so we'll focus on this one, is nostoc. Some slides you'll see Anabaina is another example of a cyanobacteria. So the challenge for cyanobacteria is nitrogenase, the enzyme that performs nitrogen fixation. It's inactivated by oxygen. We say it's oxygen sensitive. So to function, nitrogenase has to be in an anaerobic environment. So different microbes have different strategies. The strategy cyanobacteria use is they make a different cell. So I prefer the term heterocyte rather than this older term heterocyst. You can use either name on the lab exam. So cyanobacteria will make a heterocyte or heterocyst, and this is a special cell. No oxygenic photosynthesis occurs, no oxygen is being generated, and the walls of the heterocyte, or the boundaries of the heterocyte, help prevent oxygen from leaking in. So inside the heterocyte is an anaerobic environment, and that's where the nitrogenase can function, performing nitrogen fixation. Now, as we mentioned, most organisms can't use molecular nitrogen as a source of nitrogen to make amino acids, proteins, nucleic acids, and other nitrogen-containing organic molecules. But organisms can use ammonia. So the cyanobacteria are taking the inusable nitrogen and fixing it into a form that organisms can use. And again, this is an important part of the nitrogen cycle. Now, another really cool bacterium that can carry out nitrogen fixation is a soil bacterium called Rhizobium. This is a new bacterium, we haven't talked about it yet. Rhizobium live in the soil, and they have a special symbiotic relationship with plants called legumes. And legumes include peas, beans, clover, alfalfa, lupin are legumes. So the rhizobium lives in the soil, and it invades the plant roots and triggers the plant roots to form these really cool little bumps, these anaerobic bumps on the roots called root nodules. So within the anaerobic root nodules, the rhizobium can live, and they make nitrogenase and thus can carry out nitrogen fixation within the root nodules. Now, this is a symbiosis called mutualism, win-win, both partners win. So the rhizobium is taking unusable molecular nitrogen, converting it into ammonia that the plant can then use as a source of nitrogen for amino acids, proteins, and nucleic acids. But then we might say, well, okay, well, how is the plant helping the rhizobium? Well, the plant, of course, up above the ground, is carrying out oxygenic photosynthesis. It's making organic molecules like glucose from inorganic carbon dioxide and water. So the plant can feed sugars to rhizobium, and that's really important because nitrogen fixation takes a lot of energy. And so the rhizobium is going to need lots of sugar to make the ATP to drive nitrogen fixation. So we call this symbiosis, it's win-win, and win-win symbiosis we call mutualism. Okay, let's see. Let me see if I can fast forward here a little bit. Okay. And this is just a big overview of nitrogen fixation in Rhizobium. And here's just, these are so cool, you guys, these really neat root nodules on the legumes. I brought in some beans from our garden, and a lot of the bean plants, because unfortunately when you take the plants and the roots out of the ground, sometimes the root nodules come off, And I don't think my bean plants had many root nodules to start with, but there's one bean plant in the lab, in the back of the lab, and if it's in water, so if you hold up the container of water to the light, you can see a few little tiny root nodules on the roots of the bean plants. And again, that's where the rhizobium live, make nitrogenase, carry out nitrogen fixation. And this is just a cartoon, folks, of a stained thin section through one of these root nodules, and these are all the bacteria inside the root nodule cells. And again, just some chemical crosstalk dialogue going on between the rhizobium and the plants. Okay, so that was nitrogen fixation. So the second topic for this unit, you guys, is biofilm formation, which we have talked about in lecture. So this is just a cartoon showing the steps in biofilm formation. And one of the concerns with biofilms that form in our patients, if the biofilms contain pathogenic bacteria, it's really hard to kill them. So why are bacteria living in biofilms so much harder to kill? Well, there's several reasons. Sometimes a biofilm gets really thick, and so the layers become a diffusion barrier for antibodies, antibiotics. Our phagocytic cells can't reach the bacteria, cannot attach to the bacteria in the biofilm, so our phagocytes can't kill them. Often the bacteria in the lower layers of the biofilm are in what's called a stationary phase of growth, and when they're in this kind of resting stage, they're naturally more resistant to antibiotics. So this is a huge problem if you have pathogenic bacteria living in a biofilm in your patient. The biofilms often form on either damaged tissues or artificial surfaces like indwelling catheters, or say an artificial joint, or maybe artificial heart valves. And in some cases, you can't kill the pathogenic bacteria, so you have to go in surgically sometimes and remove the artificial device, treat with high-level antibiotics, and then sometimes the patient has to undergo, for example, another surgery to have, say it's an artificial joint, to have that artificial joint replaced. So this can be really tough on the patient. This is just showing Pseudomonas, and Pseudomonas is notorious for forming biofilms in the lungs of patients with cystic fibrosis. And again, the Pseudomonas, we'll learn later on, they're just naturally really resistant to antibiotics because they're gram-negative bacteria, and their outer membrane porins will prevent a lot of antibiotics from crossing. So they're naturally hard to treat. And then if they live in a biofilm in the lungs, it makes them really tough to treat. This is another example. This kind of dates back to the toxic shock syndrome, when women were being sold on these tampons that you didn't have to change very often, which was horrible, a horrible like selling point. Because what happens with tampons, if they're left in too long, bacteria can form biofilms, and this is really dangerous if you have the Staphylococcus aureus make a special toxin called toxic shock syndrome toxin. Because the bacteria are colonizing the tampon, it's not being changed. So the bacteria are just pumping out these toxins, and as a result, several women died. So there was a big campaign to get these tampons removed from the market. Again, another example of how sometimes us humans, we kind of start, I don't know if you'd say we're kind of playing with nature. That's probably not a good, that's really not appropriate. But in this case, for example, with having these, you don't have to change them very often, tampons is not a good idea. And again, folks, this is a catheter, an IV catheter. Great places for biofilms to form. I've read that Staph aureus can form a biofilm on an IV catheter within like 24 hours. That's just bad news. And I'll just let you guys read the catheter-related bloodstream infections, but these catheters are just, they're just wonderful places for biofilms to form. And again, the bacteria living in the biofilm are going to be really resistant to antibiotic therapy. and more on IV catheters. Okay, so I'm going to let you all finish the biofilm. In lab, what we'll have you looking at your tooth plaque that you gram stained. So remember, the tooth plaque is a biofilm. The early colonizers, for example, are Streptococcus mutans. If we eat a lot of sugar, the Streptococcus mutans make that sticky slime layer. They stick to our teeth. They grow and divide. Other microbes can stick into that sticky layer, so you often have a polymicrobial biofilm formed in the mouth. If we eat more and more sugar, the bacteria ferment the sugar to acids, which cause the teeth to decalcify, leading to dental caries or cavities. And long term, those bacteria can cause chronic inflammation of the gums and lead to periodontal disease. And studies have shown that, furthermore, people that have really bad oral health are at higher risk for some cardiovascular diseases. It's thought that the bacteria can enter into the bloodstream, and this happens when we eat or floss or brush our teeth. And the bacteria perhaps can cause inflammation in our blood vessels, again, increasing the risk for cardiovascular disease. And then Nina Howard shared with us a bonus extra credit poster where in her hospital they discovered that in the hospitalized patients, the frequency with which, like, they were brushing their teeth, and sometimes you have to have help doing that. If they weren't brushing their teeth, it led to an increase in the risk of hospital-acquired pneumonia, and this is fascinating. So they did a study, and they discovered by increasing the number of times that the hospitalized patients had their teeth brushed, it reduced hospital-acquired pneumonia by like 60%. That is incredible. That is just amazing. So Nina's poster, we have it down in the hallway. It explains how there's micro aspirations, like when we're sleeping, we're aspirating some of the secretions in our mouth, and that's where the bacteria are. And if you're hospitalized and sick, you might be at higher risk for lung infection, pneumonia caused by these micro aspirations. Fascinating study, and a great poster if you have a chance to read it down in the hallway on the wooden table. Okay, you guys, so I am going to try to keep this short, so I'm going to end it here, and we'll try to get this we'll try to get it to YouTube and then post it, okay?